Thank you for joining this webinar today. Today, we'll be presenting a presentation that I gave at American Society of Microbiology Microbe Conference in June of 2026. This presentation is entitled Casting a Wider Net: Why Hybridization Capture is the Missing Link for PacBio HiFi 16S Surveys. So first, a little bit about myself. My name is Megan Beaudry. I'm a technical specialist at Dicel Arbor Biosciences. I have over 8 years of experience in genomics, sequencing technologies, and microbiology. I got my PhD at the University of Georgia and my Master's of Science at the University of Alberta, both in the College of Public Health. For a fun fact, I was a former Arbor Biosciences customer before I worked here. All right, let's set the stage. What problem are we actually trying to solve? Well, in microbiome sequencing, we often have complex environmental or host-associated samples, where our target 16S sequence often represents a very small part of the total DNA haystack. So essentially, we're searching for a needle in a genomic haystack. Now, how do we usually search the haystack? Well, one approach is through shotgun metagenomics, where you sequence the entire haystack. However, it's expensive, it doesn't resolve rare sequences, and most of your budget goes into sequencing the straw or non-target DNA. Another approach for 16S specifically is 16S amplicon sequencing. In this approach, you use primers to pull the needle out, but the primers have the potential to bias abundance estimates. The primers are also picky, so if the needle is slightly different, your magnet may miss it entirely. And so you may end up feeling like Mr. Microbe here, who's very stressed trying to find all of the rare sequences in his DNA haystack. And so this is where a third tool comes into play. And this tool is called hybridization capture. So instead of PCR, we're going to use bioattenuated probes as high affinity magnets to selectively fish out the target sequences from our NGS library. It's selective in that it pulls targets from complex or low biomass DNA. It's universal in that there's no reliance on degenerate amplicon primers. And it's broad, meaning it has a high tolerance for phylogenetic divergence. So you may be wondering, well, how does this work? How do we find the needle in the haystack? Well, MyBaits are a form of target enrichment that uses bioattenuated single-stranded RNA capture probes. We start with an NGS library that is denatured and reverse complemented baits hybridized to overnight. Bait target hybrids are bound to streptavidin-coated magnetic beads and pulled out of suspension with a magnet. Following this, most of the non-target DNA is washed away. Lastly, we take that remaining enriched library, amplify it, and take it into sequencing. Now, one of the key advantages over amplicon sequencing is how capture handles sequence divergence. So we know that both amplicon sequencing and hybridization capture can effectively enrich for known sequence regions. But only hybridization capture can retrieve sequences that have significant rearrangements, mutations, or novel strains that diverge from the reference. And this is because probes can tolerate 5 to 10% sequence divergence from the target without affecting the capture reaction. Our probes are also designed to overlap across the target, so adjacent probes can help grab that sequence. So how well do we actually empty that haystack? Well, we find that 16S capture has between 400 and 1200x enrichment. So this tells us that routine capture really concentrates your 16S signal, allowing for deeper, more representative profiles with less sequencing. Capture also retrieves rare taxa that are often lost in shotgun noise or missed by amplicon primer sets, providing a more accurate snapshot of that true community. And as you can see here, Mr. Microbe is quite happy that he was able to find his rare sequences. That's what his little bucket is filled with. So how can we make 16S capture even better? Well, we compare it with long-read sequencing. So combining MyBaits with PacBio HiFi delivers the gold standard of 16S data. What we have here is the result from taking a mock community of a variety of pathogens and spiking them into a complex background matrix, to kind of mimic what you may see in a real-world sample. We took these PacBio HiFi reads and aligned them to the full length of the 16S. Now I want to point out three main things here. First, we're able to get single base resolution. Second, we're able to pull down the full length of the 16S rRNA. So not just a hypervariable region or two. We're able to get all 9 hypervariable regions. Third, we're able to capture those flanking regions as well. And in a few slides, this is going to become increasingly important. So we took that same mock community that we spiked into a complex sample, and we wanted to look at the performance across all of those species in that mock community. So what we have here is a chart that shows read length on the x-axis and the number of reads on the y-axis. As you can see, our average read length is approximately 2 kb, and we're capturing the 16S, which is approximately 1.5 kb in length. So this tells us that on average, we're always able to capture some of that flanking region. However, I want to point out that longer inserts are absolutely achievable. If you have a higher quality DNA sample, capture can pull down fragments between 6 and 10 kb in length without issue. Second, what I want to point out is that we are able to capture the full length of the 16S and those flanking regions across multiple species in a single sample. And so these flanking regions are really important as they can help us resolve multi-copy confusion. So as we all know, most bacteria carry multiple copies of the 16S. Now, amplicon sequencing often collapses these, but capture can help us resolve them. So here's an example with E. coli K12, which has 7 copies of the 16S rRNA. We're able to successfully map all 7 copies in this circular genome. So by capturing those flanking sequences, we're able to distinguish identical genes based on their chromosomal location. And so this is what gives us individual copy level resolution. So to summarize what we've just talked about, we essentially have the needle, the thread, and the story. The needle is the 16S rRNA gene itself. Probes bind to regions to help pull down the entire gene. The thread is our long insert libraries, which can range from 2 to 10 kb in length, as capture can pull down the gene plus several kb of the flanking regions. And our story is that you don't just see the 16S, you see the entire genomic neighborhood. And this is the key to strain level identification and single nucleotide resolution. So there are several advantages to 16S MyBaits capture. The first is that it's a really robust strategy, making it cost-effective for deep microbiome surveys. Second, it's precise in that it resolves rare taxa and multi-copy genes, like I just showed you. Third, it's contextual. By recovering those flanking loci, we're able to get true strain level discrimination. And lastly, it's versatile. One 16S MyBaits capture kit can be used for both short and long read sequencing, like PacBio. Now you may be wondering, how do I get to use this awesome kit in my lab? Well, our 16S capture kit is a catalog kit, meaning it's ready to ship. However, we have many other catalog options to select from, which I'll talk about shortly. Now, if we don't have a catalog option that fits your needs, we also specialize in making custom capture kits. that can be completely specific to your research goals and can be for absolutely any target. For this case, you own the design and get to decide what to do with it, and you'd work with our technical team to help create that custom design. Lastly, for microbial applications, we offer what we call express panels. These panels have design parameters that are already selected. These would then be released and shared with the broader scientific community to help propel science and research forward. So, to our catalog offerings, we recently launched our new microbial collection, which offers many ready to ship solutions for bacteria, viruses, and protozoa. You can easily access this full list on our website, and there's also a scrolling list on the side of this slide. But I want to make sure to point out that catalog offerings will be continued to be added very regularly. Also, if you don't see what you need, we can absolutely make it for you. So not only do we make custom and have catalog kits that can be used in your lab, but we also have our own MyRead service lab. Our service lab specializes in wet lab services and dry lab services. So our laboratory services include things like DNA and RNA extraction, library preparation for short and long read sequencing, target capture, of course, and we have both short and long read sequencers in-house. We also offer a variety of bioinformatics services, including QC and adapter trimming, variant calling, and custom analyses and reports. So let's wrap up. What are the advantages of MyBait's hybridization capture? Well, MyBait's works well with highly degraded DNA. It tolerates unknown polymorphisms and recovers those flanking loci like we talked about earlier. Additionally, deep sequencing is not necessary to get the required coverage. There's also no limit on the target size, and you can use it with any NGS platform. Additionally, all our kits contain all the reagents necessary for the capture assay. Finally, MyBates has helped power over 1,300 published research studies to date. I want to thank you for listening to this webinar. Please don't hesitate to reach out to me. My email is at the bottom of this slide if you have any questions, or to our sales team whose email is also on this slide. Thank you.